all animals need to provide energy for themselves, right? This energy allows us as humans to run, to do daily things, for our heart to beat, allows us to breathe, right? All of these things require energy, okay? We get that energy by eating, right? And whenever we eat, we're really giving our body a number of different biological molecules. And these very commonly fall into some large classes. And these are things that you've heard before. So lipids, which is another word for fats, proteins, sugars, and we could list more, but let's go ahead and stop there. And as it turns out, nearly all of these molecules are, in some way or another, adopt this kind of beads on a chain overall structure. We call these polymers. Okay, so this would be an example of a polymer. Polymers are constructed of small molecules linked together through easily formable and easily destroyable bonds. So each of these bonds that are linking together these individual sugar units or individual fatty acids or individual amino acids, they're all linked together through bonds that are fairly easy to break. And that's really important because if we're going to use these molecules as a source of energy, we need to be able to break them down into these individual pieces so that we can metabolize them to produce energy. Okay, so in the case of lipids, in the case of fats, this bond that's formed is an ester. Okay, so we know that esters are some sort of hydrocarbon chain, which I'm going to go ahead and just abbreviate as an R here. Okay, R, it could be any number of different hydrocarbon chains. So this R is linked together with another R group through an ester linkage. And let's make the other R group a different color. So here we have R prime indicating this is a different carbon chain. Okay, so we build up large molecules of fat and they're linked together through this ester linkage. So when we metabolize these fatty acids, we need to be able to break this ester linkage to use the individual fats as food, as energy. Okay, the class of chemical reaction that occurs is known as a hydrolysis. Okay, as the name suggests, it involves water, and this lysis means we're breaking. So we are going to break a chemical bond by using water. And the way that this works is I'm going to go ahead and redraw this molecule here. So we have our ester linkage, and attached to our other R group, we react with water. And when this reaction occurs, we form two new molecules. So in this case, I form what now becomes a carboxylic acid. And the other compound I form with my other R group, so R prime here, we end up as an alcohol. And I'm trying very hard to color code these things so you can see where the atoms come from and where they go to. So in this case, everything in yellow was water, right? So our water is here. What we observe is that the two hydrogens and oxygen from water end up split apart. So water splits to create part of the carboxylic acid and part of the alcohol functional groups that are produced. We see that this ester linkage gets split right down the middle. So this bond between the carbonyl of our ester and the oxygen of our ester gets broken. Okay, and we reform two new bonds. This is a hydrolysis reaction. And what you need to get good at is looking at a compound that contains either an ester or an amide and predicting the product of a hydrolysis reaction. So what I'm going to do now is go ahead and kind of show you an easy way to do that. Okay, so we mentioned that we're dealing either with esters or amides. Okay, so here I have an R with my carbonyl, and if this is an ester, I have OR prime. Okay, I'm going to put in my lone pairs here. And we're reacting with water. Okay, so what you want to do is line the water up where the oxygen lines up with the carbon of the carbonyl, and this hydrogen lines up with the oxygen of the ester. Okay, and our other hydrogen hangs out over here. I'm going to go ahead and put in my lone pairs here. So now what ends up happening, so the way you can think about this is every time you break a bond, you need to make a bond. So if an atom has a bond that's broken, it needs to make a new bond. What we know happens in hydrolysis reactions is that we break this bond between the carbonyl carbon and the oxygen. So this bond gets broken. Okay? So this oxygen right here has a bond broken to it, meaning we need to make a new bond. And it's going to make a new bond, so this pair of electrons end up making a bond with this hydrogen. Okay? This hydrogen has a new bond made now, so we need to break a bond. We break this bond. This oxygen has a bond that's broken, so we need to make a bond, and we make it with this carbonyl carbon. So you're basically making a square. We know for sure that this CO bond has to break, right? So this bond right here must break. So this carbonyl has a bond broken, we make a bond. Every time you make a bond, you break a bond. So the product of this is going to be R attached to the carbonyl, but this carbonyl is now attached to this OH. So we create a carboxylic acid, and this R prime is now attached to an alcohol. So it's always true that the product of hydrolysis of an ester will give you a carboxylic acid, and it will give you an alcohol. Okay, let's see what it looks like for an amide. So for our amide, we have R, we have our carbonyl attached to a nitrogen, hydrogen, and this nitrogen attached to an R prime. Okay, so water comes in. Water, the oxygen gets lined up with this carbonyl, the hydrogen gets lined up with the nitrogen. So here's our water. Okay, so we know we have to break this bond. So we're going to make a bond here, break a bond, make a bond. So we created a square. So we end up forming a carboxylic acid just like we did before. So I have R carbonyl, OH from water. So here we have a carboxylic acid. And the other molecule we form is R prime attached to this NH2. So we create an amine. So we have R prime attached to NH2. So we create an amine. Every single time you hydrolyze an ester, you create a carboxylic acid and an alcohol. Every single time you hydrolyze an amide, you create a carboxylic acid and an amine. Okay, and these are really the only types of reactions that we're going to think about hydrolysis for. So if you're comfortable looking at an ester and an amide and predicting the products of hydrolysis, then you're in very good shape. In the next video, we are going to explore the energetics of these reactions, and then we're going to begin thinking about how we can avoid the really large activation energy that we're going to predict is necessary.